So today I'm going to talk, just like Megan did, on the relationship between the microbiome early in life and asthma. And given the topic today of health disparity, I thought that I should introduce asthma with a slide on just that. So asthma is an extremely common disease. There's over 400 million people that suffer from asthma. It's the most common pediatric immune chronic disorder in the world, but it doesn't affect different populations in the same way. As you can see, the distribution is disproportionate. In red, you will see the nations that have the highest prevalence in the world, including many industrialized nations like Canada, Australia, the US, the UK, some places in Europe. But despite what some studies or papers say, asthma is not really only a disease of industrialized countries. And in fact, some countries from South America, they show some of the highest incidence of asthma in the world. But there's an interesting difference there. In most of the industrialized countries, there has been a really fast and steep increase in the rate of asthma. And in places like Canada and Australia, the incidence of asthma has quadrupled in only 25 years. That has not been the case in, for example, Peru or Brazil. Asthma has been high for a longer time. And what that tells me, or what that tells, is that there may be different factors, potentially environmental factors, that are driving this. But as I said before, asthma is an extremely common disease. So the second thing that I'm going to tell you is that it is extremely easy to develop asthma if you compare it to other immune diseases like autoimmune diabetes or other immune diseases. It is just really easy to get asthma. And just as an example of how easy it is to get asthma, I'll show you how easy it is to give asthma, or what we call asthma, in a mouse. And I've worked with other models of immune diseases before, given autoimmune diabetes or IBD, and you'll see how easy it is. So these are two very common models of asthma. The first one is the Ova model, and the second one is the house dust mite model. For the Ova model, what you do is that you take a conventional animal, it doesn't have any genetic predisposition or anything, and you will give two systematic stimulations of the immune system with Ova, which is an antigen that the animal has never seen before. Then you wait for a couple of weeks, and then you do a series of intranasal challenges with the same antigen. So you just make the animal inhale this antigen, and that's it. It will provoke a chronic inflammatory response very similar to human asthma that would last for a long time if you keep exposing with the antigen. The house dust mite is even simpler. You even bypass the systemic immune response, and you only give the antigen by inhalation a couple of times, 2 weeks apart. And then a week later, 2 weeks later, you do a series of inhalations again, and you provoke the same disease. So it's not that big of a surprise to understand why asthma is so prevalent in the human population. And what this tells us also is that, yes, asthma, just like many other chronic immune diseases, it's really a combination of genetic factors and environmental factors. But with asthma, as opposed to other diseases, it's really susceptible to these environmental factors. And I think now in the microbiome era, we really have to, like Chris was saying, increase or rethink what we understand as environmental factors. The epidemiology of asthma is very telling as well, and Megan did a really good job at explaining some of these. So I'm just going to go over a couple of them that I think were not explained perhaps. One of them is farm. So if you are raised in a farming environment, your risk of developing asthma is much lower and is one of the strongest factors. She mentioned the other ones. One that was not mentioned was helminthic infections. And now the data with helminthic infections is really strong when that factor has been taken into account in the studies, but it really changes. So it really changes with the type of infection that you had, and it also really changes to the place or the country where this study took place. But what all of these factors really tell us is that these exposures early in life, and all of them can be related to the microbiome, have a very strong role in asthma development. So when we think of that, we have to think about the early life microbiome. So a couple of things about the early life microbiome, even though a few of you have talked about it before. This community of microbes goes through some very drastic changes during the first 6 months of life. We're born sterile, but by the time we're only a few days of age, we're completely colonized with microbes. And what follows in the next few weeks and months is a series of ecological shifts that last for about 2 or 3 years in most human populations that have been looked at. But specifically, the first 6 months of age is this phase that experiences this larger shift. And what one has to remember is that as this community is changing, the human is undergoing very large changes in immune development as well. And when you think in ecological terms, when a community, whether it's microbes or large animals, is establishing itself or is changing, it is inherently low in resilience. And this is happening at the time, a critical time in terms of immune developmental stage. So we do know through lots of really important work in immunology that the immune system, not just in humans but in other species, is especially susceptible and permissive to microbial signals during this stage. So what is the evidence, and I'll provide a few here, what is the evidence that early life changes in microbiome actually affects asthma development? The first body of work came from animal studies, of course, and this is a PhD student in Brett Finley's lab where I did my postdoc some years ago, and she just had a really important but very simple question. She wanted to see whether if you gave antibiotics early in life versus later in life, if that would change the susceptibility to asthma. So she did just that. She gave those mice either streptomycin or vancomycin early on, and then she ran the Ova model of asthma that I explained before. And she had 2 groups. She had a group of animals that were giving either antibiotic from birth on, or animals that were given antibiotics when they had become adults or after 7 weeks of age. And very briefly, what this report showed was that only those animals that received the neonatal exposure from birth with vancomycin showed an increased susceptibility of asthma later in life. That did not happen when the same antibiotics were given in adult life. And this, on the bottom here, this was characterized with what you would expect in this type of allergic immune response, which is eosinophilia and antigen-specific IgE. The same year, and this came out of David Ardis's lab, they did the same thing. So here you have animals, young animals, that were either conventional or treated with very, very high doses of antibiotics. And that showed that there was an increase in IgE and in basophils as well. They proposed that at least one of the mechanisms, immune mechanisms that is in play here, and this is what you would consider a healthy microbiota, and over there is antibiotic-treated or depleted microbiota, that in the absence of these signals, you're going to induce or favor an overproduction of IgE, and this IgE is going to have effects at the level of the bone marrow that are going to lead to expansion of basophils, and that is going to create a Th2 or allergic immune milieu, if you may. Now, we were interested in trying to see if there were some evidence of this in humans. So, and Megan did an excellent job at introducing the child study for me. We were really, really fortunate that we were able to collaborate with the child study, and we received a sub-cohort of these 3,500 samples that were collected, about 320. And we randomized them according to their symptomatology at 1 year of age. So we had 74 healthy controls, and we had 87 kids at 1 year that were atopic, and that was considered with a positive allergen skin prick test. And then we had wheezers, and these were 136 kids that had at least one episode of wheezing. And then in the middle there, we had 22 of these subjects that had a combination of both atopy and wheezing. There's a number of predictive indices that can, of course, predict the risk of asthma based on these symptomatology at 1 year of age, or at 2 or 3. And what one of these indices, the most stringent of them, was able to show that these 22 atopic wheezers were at a significantly higher risk of developing asthma by the time they started school. So we had 2 samples at that point. We had a 3-month-old sample and a 1-month sample of these babies. And at that point, this was circa, I think, 2012, which seems like a generation ago in terms of microbiome and sequencing technology. What we did was 16S analysis on it. And what I'm showing here is the confirmation via qPCR of what we observed bioinformatically. We didn't see any broad changes in community ecology, whether it was alpha or beta diversity, but we did see some significant changes at the OTU level that we confirmed via qPCR. So what we saw in these babies at 3 months of age was a significant decrease in these taxa, Veillonella, Lachnospara, Rothia, and Faecalibacterium. We included Bifidobacterium there as a way of a control because we hadn't seen any changes from the data set. When we looked at the 1-year data set, we found that the differences in the first 2 taxa were less evident than what they were at 3 months of age, and they had disappeared for Rothia and Faecalibacterium, suggesting that perhaps it's those changes that happen in the first 3 months of life, the ones that we can associate with asthma later on. Now, we were interested not only in who these bacteria were, but what are they doing. I don't need to introduce short chain fatty acids to you, so I'll just say that this is what we measured. And what we found in these babies at 3 months of age was that there was a significant reduction in acetate. We were also really fortunate that the child study had collected urine from these babies, which is not really easy to do in these types of studies. So, but they did, and we were able to obtain urine samples from the same babies, and we sent them for metabolomic analysis. And what we found actually quite a strong signal in terms of metabolites associated with later asthma risk. But we found some of them that we can directly correlate with bacterial metabolism or gut bacterial metabolism that were significantly different between babies that were at risk of asthma or the controls. The ones in blue are related to secondary bile acid metabolism, but there's urobilinogen that is associated with hemoglobin metabolism. And we still, of course, do not know how this may or may not be associated with asthma development. But what we do know is that these could very well become biomarkers that precede asthma really, really early before babies start wheezing or developing atopy. Now, those findings were really interesting and novel at the time, but they really remained an association. And most studies, I would argue, that's what they show, interesting associations. So we decided to go one step further and try to show whether there was a causation association here in mice, and we used germ-free mice for this. And the experiment was actually pretty simple. We had pairs of breeding mice, germ-free, and we had groups of them that were given what we call an asthmatic microbiota. So this was fecal microbiome from a 3-month-old baby that was later formally diagnosed with asthma, but importantly had really low levels of these 4 bugs that we had found in the microbiome study. And then the other group of mice received the same inoculum, but we grew 1 species of each one of these taxa, and we supplemented the inoculum with that. And then we bred them. We continued the inoculum for another 3 weeks, and then at 7 weeks of age, we did the Ova model of asthma. And what we found was that by changing the inoculum so boldly, we were able to really shift the microbiome of these mice. When we were looking at the 4 taxa that we were able to grow, we especially for Lachnospara, they were able to colonize really effectively the mouse gut. So the shift that we saw in the mice was was very big, which was definitely not the case as we had seen in humans. But it allowed us to show that if you change the microbiome with these particular four bugs early on, you can really change the outcome of this immune disease later on. So animals that were given the asthmatic microbiome with these flavor bugs, as we call them, we saw an important reduction in the inflammatory response. And as you can see in the histopathology over there, the image here in the bottom was a lot less inflamed than the animals that receive the asthmatic microbiome. And this was really interesting because asthma really, we understand it that we study asthma as a non-communicable or a sterile disease. But here we are just changing and modifying the gut microbes that an animal is colonized with. And that has a drastic change in an immune disease later on. Now, what about asthma elsewhere? And as I introduced before, the global distribution of asthma is pretty disproportionate, but there are some countries in South America that have a very high incidence. And we were very fortunate to partner up with Professor Phil Cooper, who has been studying asthma in Ecuador for a couple of decades now. His interests, as of late, had been to try and understand the relationship between helminthic parasites and asthma. And because of that, he had run this great longitudinal prospective study where he collected tons of fecal samples that were just sitting there in a freezer, and that usually never happens. So it was a great collaboration with Phil. So a couple of things about the town in Ecuador. So Ecuador is right here. The town is called Las Esmeraldas. It's a coastal town. Socioeconomically, is very underdeveloped. And the incidence of asthma, I know this is really small, but it's about 11%, which is very, very comparable to the incidence of asthma in Canada. We had almost 100 children for which we had samples at 3 months of age, and we randomized them in the same way as with the child study. So we had the atopic wheezers, the kids that develop atopic wheeze later on, or the controls. Now, even though the incidence of asthma is very similar in Ecuador than in Canada, the environment is definitely not the same. And what's particularly interesting about this population in Las Esmeraldas in Ecuador is that they have experienced a very quick, like a 1 generation long, rural to urban transition. So only about 15 years ago, most of the homes in Las Esmeraldas, as Phil tells us, they look like that. They were built in very close proximity to the jungle. So the type of environmental exposure just on that was really, really different. Many of the homes didn't have windows, didn't have proper flooring or roofing, and through governmental public policy changes, they started housing developments for a really large proportion of this and other areas in Ecuador. And now, about half of the people that used to live in this type of environment lives in an environment like that. So that on its own is a really big change. What he's been able to measure over the years is that is also associated with an increase in allergic disorders, including asthma. If we look at the epidemiology of this sub-cohort that we analyzed, this on its own is really telling. So there were some things that we had found in other cohorts before. For example, the use of antibiotics during pregnancy increased the risk of developing asthma, whereas vaginal birth decreased it. Breastfeeding, although more conservatively, but it decreased it as well. Helminths, of course, appear here because these areas are endemic for helminthic infections. So infection with Ascaris increased the risk, but infection with Trichuris decreased the risk of developing asthma. Another one that commonly appears in asthma literature is respiratory infections. The higher the number of respiratory infections that you have during childhood, that also increases the risk of developing asthma. However, there was one that to me was really, really interesting, just for the fact that we could measure it. About half of the households in this town did not have a source of potable water at home, so that allowed us to measure that. And lo and behold, if the individuals had potable water at home, that increased the risk of developing asthma. And of course, it's only an association, but it's a really interesting one, and it's one that is very much in line with what was originally proposed in the hygiene hypothesis. Now, this was, of course, a microbiome study. At that point, we ran 16S using MySeq, not HiSeq as before with that child study. And we found some changes at the OTU level for bacteria. Interestingly, most of those changes were different than the ones that we had found in the Canadian cohort. But that, of course, in a way should not be surprising, considering that both populations were expected to have different microbiomes to begin with.
I just want to ask one question. Can you just quickly go back to the previous slide just for a second? Yeah. Did the households with the non-potable water, were those the places primarily where you found the asterisk and the trichrist? I
do not know. I don't think they recorded that date. We could look at it that way, but that would be interesting to see. Yeah, and one of the original hypotheses that we have, not necessarily, it could be potentially associated with Helminth, it's the fact that these children may have more diarrheic events, and that on its own is going to change the immune development in a way that makes asthma or allergic disease less likely to occur or develop.
Also, I think asterisk can migrate through the lungs as well. Which,
yes.
Unlike other parasites, it might be one of the things predisposing.
Yeah, and that's another interesting thing about the literature in Helminth. If the Helminth has a systemic cycle, that may have a very different immune consequence than if the Helminth stays in the gut. That's correct. So going back to this, the dysbiosis that we found in this cohort was different than the dysbiosis that we found in Canada. But when we measure acetate, which I should remind you was decreased in the Canadian cohort, we found the same thing. It's also decreased in the Ecuadorian 3-month-old children, perhaps suggesting that if we're going to look at trying to find a biomarker, taxa may not be the best way to go given the inter-individual and inter-population variability, but that metabolites may be a better option. Now, the next thing was really a surprise for me. At that point, because we knew that this population has endemic levels of parasites, we decided to do an 18S screen as well. And 18S will screen for all things that are not prokaryote or archaea. And what we found was that at 3 months of age, we didn't find any helminths or protozoans, but we found was a ton of fungi. And these are the fungal OTUs that were associated with asthma risk later on, more than double of the bacterial ones that we had found. Especially this one here in blue, Pichia kudryavtsevi, which I've never heard of before in my life. Before this is a yeast somewhat closely associated with Saccharomyces and Candida. And we confirmed that via qPCR. There really was an important increase in this yeast in the majority of kids that went on to develop asthma. The other thing that struck me when I was analyzing the 18S data set was that in proportion to other eukaryote sequences, the fungal sequences belonging to the atopic wheezers seem to have a higher proportion. That appeared to be significant, but we decided to use a qPCR as a way to try and confirm that. And it seems, although it's only 1 star, it's not that significant, that there's more fungal specific sequences in the babies that go on to develop atopic wheeze later on. And this is something that we want to confirm now in the child study and in other animal studies that I'm doing now. So for those 2 studies in 2 very different places that call asthma the same name, we found that yes, for sure, there is a dysbiotic microbiome signature that can be associated with later risk of asthma It's different in terms of bacteria. For the Ecuador, we were able to find fungi as well. We're in the process of looking at it in the bacterial kids as well, but it seems that the fungal signal is even more correlated with that. But what is interesting with this type of work is that potentially we're looking at a time in life, which is 3 months, but I could argue it could be even earlier than that, where we could really discriminate what can happen with this child later on. As I said before, it may be better to focus on microbial-derived metabolites when we looked at things like biomarkers. Now, the other thing that we did, I was really intrigued with this fungal finding. I was not expecting it by any means, but it got me thinking. Fungi could be, Ecuador or what could it be? And it occurred to us that potentially it had to do with antibiotic exposure. So we know that antibiotic is really going to kill a whole bunch of bacteria, and potentially some fungi are going to take advantage and are going to bloom at least temporarily, and that may have an immune consequence. So what we had in this cohort was information on the duration of antibiotic use. So we correlated that with the abundance of the 18S OTUs. And we found significant correlations between Aspergillus niger and Pichia kudryavtsevi. And as you remember, Pichia was also associated with asthma. And that allowed us to develop this hypothesis that perhaps it's not just bacteria, but that antibiotic-driven alterations in the fungal intestinal microbiome may affect immune development and make some individuals more susceptible to asthma. And this is some of the things that I'm doing now. This data, of course, is very preliminary. It's unpublished. But one of the first approaches that we took before we could get our germ-free animal facility up and going was to study this in conventional mice, and we had 3 groups of mice. One group was just treated with antibiotics, and this was Augmentin, which is a very common one given to children under 6 months of age. The other group was given antibiotics with 2 yeast species, Candida albicans, which was also showed in a different cohort to be associated with later asthma risk, and Pichia kudryavtsevi, which is what we found in the Ecuadorian kids. And then the other group was just untreated. And then we gave Ova and alum intraperitoneally a couple of times before we assessed just the general immune system in these mice. The first thing that we found was that it was only in the group that received the yeast with the antibiotics that we were able to see more fungi, and this is by culture. One of the nice things about now measuring fungi is that they grow really, really well, even outside the anaerobic hood. So it's a lot easier because you don't have to wait for sequencing results to get back to you. But then when we looked at their systemic immune response, and this is a splenocyte culture here at 11 weeks of age, the group that received yeasts early on, for 1 week early on, had a lower proportion of IL-10 positive cells. IL-10 is a very important cytokine in the regulation or the dampening of the immune response, and also an increase in IL-13 and IL-5, perhaps also IL-6 and IL-4, all of which belong to this Th2 or allergic immune response. The other thing that we wanted to answer, and this is the one line of work that we're developing more and more now, is to just ask a very basic question that up until now hadn't been asked in germ-free models, and is what happens with the immune system when you colonize the gut with fungi? All of these studies have been done in association with bacteria only, but not fungi. So very simple question, and we did this in collaboration with Kathy McCoy, who is a very well-established mucosal immunologist that also happened to open a brand new germ facility in our institution. So what we did was that we associated, or we colonized some germ-free mice with a mix of 12 bacteria, 12 mouse-derived bacteria that we know of. And the other group received the same 12 bacteria plus the 2 yeasts, Candida and Pichia. we were able to see that both species actually grew, and we were able to recover them even weeks after the initial exposure. The first measurement that we did was a physiological measurement, and what we found was that the transepithelial permeability of the colon in these animals is quite different, just by the fact that one is colonized with fungi and the other one isn't. So the colon of these mice colonized with fungi was leakier. We also did organ culture in colon and in the jejunum, and we found important differences in IL-10, IL-9, and TNF in colon, and interferon gamma, IL-17, IL-10, IL-17F, and TNF-alpha. So important and broad changes in the immune response only because you're colonized with a different life form. When we looked at not just at the mucosal immune response, but at the systemic immune response, again, just by colonizing with fungi, there's really important changes in the proportions of cell types or cytokine-specific cells or cytokine-producing cells in the spleen. So from the very basic proportion of B over T cells to an important reduction in Tregs and in CD8-positive T cells, and also important increases in Th2 cytokines. So I do think now, even though it surprised me at the beginning, that there is something interesting with fungi, and it's not just bacteria that is modulating the immune responses early in life, at least in the context of asthma. So in summary, I hope that I've been able to convince you. I think there's really, really strong evidence now that supports that it's not just a mere association. Changes in the early life microbiome can influence asthma development. And it seems that early life is the most critical stage for the development of the microbiome and for the influence that the microbiome has in the regulation of the immune response. It seems that the first 100 days of human life, at least from the studies that we've done, they represent a true window of opportunity during which changes in this microbial signals can have a long-lasting immune effect and can enhance the risk of asthma and other allergic diseases. It also appears that this dysbiosis will change across population, just simply because humans are colonized with different microbes in different parts of the world. At least from our equality study, this dysbiosis is not just bacterial, it is fungal. And if you can take only one thing from our work, is to potentially considering going beyond just 16S and trying to measure other life forms, because I was really surprised with what we found. And even though we haven't confirmed it, but we do think that fungal overgrowth may also explain immune alterations that lead to asthma. And right now we're doing exactly that. So we're carrying out clinical studies where we look at babies that show up to the emergency room at the hospital. They're given antibiotics, and we're trying to study what happens to their microbiome before and after the course of antibiotics, with particular attention to what happens to their fungi. And also, I think one of the most important things that can be taken from this talk is how important these prospective observational studies can be. It is really the only way that we can look at those potential changes in the microbiome that precede disease. Because if we're looking at changes in the microbiome that happen in combination with disease, it's really hard to tease out what came first, the chicken or the egg. So these studies, they're complicated and expensive to run, but they're extremely necessary. And with that, I would like to thank the people in my small lab that were in charge of some of the preliminary work that I showed, as well as the different people from my lab, in my postdoc lab with Dr. Ben Finley, as well as other collaborators and funding. Thank you.
Questions? I just want you to elaborate on one of the slides. You were really emphasizing the first 100 days as being very important. How far do you go out? I mean, in the sense of how important is it relative to some later exposures, later environments, later
Well, there's been a few, so there's these two studies that we presented here, but there's been data from other prospective studies showing earlier even time points, 2 months of age, so that the work by Susan Lynch, for example, they found that some of the most important associations happen even earlier than the 3-month mark. 100 days, based on those studies, seems to be where you find those stronger associations, but it is possible that there's smaller signals later on.
I'm just wondering, what do we know about the decay and the strength of that, like in 6 months?
Yeah, there's not a lot known. There's some animal work, but this is of course in animals only, showing that another critical period in immune development is during the introduction of solid foods, and that occurs during the month 4 to 6 in most human populations, but the human data of that is lacking. But you could speculate that that may be another important event, given that as you increase the diversity of nutritional substrates, you also increase the diversity of microbes. So that could be an important event, but it's really not known at this point how far off.
I have a question about the Helminth's trichoris infection. That seems to be the most important factor, relatively speaking, preventing.
Yes.
And so, do you think asthma increase is a loss of function or a gain of function?
I think it can be both, and it really depends on what variable you take into account. Asthma is not really one single disease. Asthma, yes, it is manifested by inflammation in the lung and constriction of the lung muscle, but the immune mechanisms that can be behind those symptoms really seems to vary inter-individually and also across populations. So my idea of asthma from a developmental point of view or a pathogenesis point of view is that there's different factors that may lead to the same consequence. So in the case of the Helminth's, it could be a loss of function, but there's other cases where asthma develops where Helminth's are not part of the picture.
Also, for you showed that the fungal overgrowth, the fungal colonization of germ-free mice, they impair the gut barrier. I'm just curious about the mucosa of the respiratory system. Do you think that part also impaired and so allow the antigen molecule to pass through?
This is one of the things that we're trying to answer now. So these are really recent experiments. We collected samples from the lung for histology. The measurements for epithelial permeability are really not easy. It's a lot easier to do in gut, which is why we did them. But perhaps there's certain things that we can look physiologically through histology to look at that as well.
It looks like with the fungi, you not only drove Th2, you drove Th1 as well, IL-6 was up. How do you explain that?
Well, IL-6, from the asthma literature, it seems to be a really important cytokine early on, at least in the animal models. You need IL-6 up for asthma to develop, the same with IL-9. So it's one of those plastic, if you may, cytokines, at least early on. It is more consider a Th1 cytokine, but it's not like TNF or interferon that usually only occurs in these cell-mediated Th1 immune responses. So for IL-6, I think what's happened, well, there's two things. In this germ-free model, what we think it's happening with fungi is that they're eliciting just a broad immune response. Most of the increases that we saw were in Th2, but we also saw close to significance increases in interferon, for example, in the spleen. So there is an overall increase, but there are some of these cytokines that early on are important in the development of a Th2, even though they're classically understood as a Th1 cytokine.
So you showed the CD3 was markedly elevated, or the whole T cell population.
Yeah.
Thank you. Thank you.